Hello everyone, how do you feel today? Happy? Very happy? Or in love? Okay, let's continue with our lessons. In this video, we are going to talk about first about the non-coding DNA and coding DNA regions. If you look at this very nice and beautiful graph, you will find out their non-protein coding sequences make up only a small fraction of the DNA of prokaryotes, as you can see over here. Oops, what happened? I have no idea. Among eukaryotes, as their complexity increases, then generally they have the proportion of their DNA that does not code for proteins. So if you look at this graph again, here is showing the percentage of DNA which is not coding. This means that in prokaryotes, most of the DNA is coded, and then the more organism is becoming complicated or more complex through multicellular organisms, then the number of the non-coding regions are increasing, and in human it reaches to the top that it has highly amount of non-coding regions comparing to the other organisms. Let's have a look at this DNA. In DNA, according to coding and non-coding regions, we have two definitions for them. One is called exons, and the other is called introns. I was thinking how you can memorize which one is more important, because it is going to be translated and it's going to be a coded or coding region versus non-coding region. What I found, the best way that you can remember, you can say exon, this X, and say, oh, this is extremely. I don't know why this line is coming out when I'm writing. Okay, extremely important, right? Extremely important. So exon, extremely important. What does that mean? This means that the exon will be kept as coding region. And intron, forget about intron. Don't think about in as in or x as x. It's more complicated. You never remember which one is going to be in, which one is going to be out. But this is the best way I think you can remember that exon are extremely important, so they will be kept and they are considered as coding regions. So what happens to intron? Introns are taken off, will be removed. So introns are removed, as you can see over here, and just exons will be attached to each other. As you can see here, only exon will be attached to each other while the introns are removed, and make what? Yes, make the mRNA, which includes only exons. So I hope you got it right, that this is the process for transcription, which means that the DNA needs to translate. And once we are talking about the coding and non-coding region, means we are talking about transcription, means making a copy of the DNA, followed by translation to make it as a polypeptide and protein. So the first step of transcription is making RNA, which is actually mRNA. But the RNA here, what's happening is making a copy from the whole part of the DNA, but this RNA will be spliced, which is called by a complex which is called spliceosome. Well, I think we have talked about that before. And that complex makes this possible. So removing the introns and just adding and pasting the exons together and makes that spliced mRNA, which is ready for moving to going out from the nucleus, and then going out from the nucleus and then move to cytoplasm and attach to the ribosomes for protein translation. When a gene is going to be transcribed, there is some areas before the gene, the gene, such as this is the DNA, and if we consider this as a gene, then in this area we have some sequences, and this is before starting a gene. This is called promoter. Promoter is the area that RNA polymerase will recognize and will attach. This is the place that RNA polymerase will attach, like here, and then start to function for making a transcription. So then it creates the RNA, which finally, after making a spliced, become mRNA. So the RNA is completely transcribing from the DNA after the RNA polymerase attaches to the promoter, which is located here. However, there are some regions in this area, not just in the promoter area, that are responsible to make this transcription happening or to stop this transcription. So those areas, let me read this together. Some of these regions act as binding site for particular proteins, which in turn affect transcription of the nearby gene. This is a nearby gene. And these are called enhancer or silencer. So what are the enhancers? Enhancers are sequences that increase the rate of transcription. So if we have enhancer here, then this means that the transcription rate will be increased. The number of the copies for RNA will be increased. That's the reason we call it enhancers. And what if it is silencers? Then silencers inhibit transcription. And when the protein is bound to this silencer area, then the transcription will be stopped. So why does this important? Is because the DNA is ordering or is leading the process for which gene will be transcribed or which gene which doesn't necessarily need to be transcribed. And this is called actually gene regulation. We will have this gene regulation topic later with much more details. But this means that which gene needs to be transcribed and which doesn't necessarily be transcribed. Let's watch two videos regarding enhancer and silencer and see if you can answer the final question at the end of the video.
So I'm a protein, and I bind the sequence tens of thousands of letters away.
And even though she's that far away from the start of the gene, it's okay because DNA can fold, as we saw just a second ago, and now her sequence is actually really close to the beginning of the gene or the Tata box region here.
So if I'm a protein that promotes transcription, what am I called?
Well, a sequence that promotes transcription here at this site would be called an enhancer. Now, not all sequences have to promote transcription. Some of them inhibit transcription, and we call those sequences silencers. Now, if we know that the effect of an enhancer or a silencer is because of whether or not the protein is bound or not, I want you to look at the following scenarios and tell me which scenario matches with which level of transcription. This could be a little tricky, so focus on each picture and see if you can match which ones belong to each other. Match the scenario on the left with the amount of transcription expected on the right. There are 3 scenarios here on the left, 3 scenarios on the right. The ones on the right are lettered A, B, and C. Just put the letter in the box that corresponds to the situation on the left where you'd expect that situation to be a match.
No. You need to pause the video and answer the question.
Through this one. Now we have 3 scenarios, a lot of I know we just learned a lot of new terminology, but hopefully you're able to breeze right through this one. Now we have 3 scenarios, a lot of transcription, a medium amount of transcription, and no transcription. If we need to match the scenario on the right with the scenario on the left. So which scenario on the left would give us, let's say, the least amount of transcription? Well, obviously if our silencer is bound, that silences transcription. Enhancer enhances transcription. So if we've silenced our transcription and we don't have anything to help enhance, this is probably the scenario in which we're going to have little to no transcription at all. The scenario above it, however, we have the enhancer bound and the silencer is unbound. Remember, the proteins mitigate the effects of what these non-coding regions can do. So a bound enhancer is going to enhance transcription, an unbound silencer won't be able to silence transcription. And so this is going to be our most active transcription here, lots of transcription, so the answer to this one is A. And that leaves this last scenario where we don't have a bound enhancer, we don't have a bound silencer. The promoter is left to initiate whatever transcription it can on its own. And so we reach the scenario of some transcription. It depends entirely on the promoter region itself, but in general, there's going to be some transcription that happens at the site. But because there's no enhancer or silencer bound, we're not going to have these extremes show up where we have a lot of transcription happening or no transcription happening.